Okay, everyone has the pH lab opened up in the Numbers app. It's been a while since we've done numbers. Remember, this is a spreadsheet. But did you know that the spreadsheet can also be a word processor? It can act a lot like Apple's Pages app. Did you also know that Apple's Pages app can also do things that a spreadsheet can do? So they've kind of intertwined their apps so that they can do similar things. But to do a lot of spreadsheet stuff, you need to be in Numbers. To do a lot of word processing, you need to be in Pages. But they allow you to really work closely together. Apple's done a great job of integrating all of their apps together. And their menu system looks very similar. So they kept it really simple and elegant. I like Apple's apps because of that. And I'm on my MacBook, and even the web version or the Mac version of this app looks somewhat similar, but the menuing's a little bit different. So not exactly what you see. So have you explored the 5 tabs at the top? That's kind of a cool thing. This is having as many spreadsheets as you want inside one file. They typically call it a workbook, and each tab is a sheet or a spreadsheet. So we're going to click into these, but just check it out. I've got a background tab, which talks about the introduction about pH. I created another tab, and I have some of our textbook information right here. So this is chapter 2, section 14 in our book that talks about pH. We're going to read some of this together here in a minute. Then it has the procedural steps that you're going to do in today's lab. Don't forget to put your name right here. So maybe you want to do that right now so you don't forget. I put it in red so you wouldn't forget. So go ahead and type your name right there. You're going to convert all of this into a PDF file. So scroll down here on the procedure, and it talks about how we're going to run the lab 1 through 4. And you're going to make a data chart in number 5, and you're going to answer conclusion questions. And this really should be number 7. You're going to turn this in as a PDF file. We've done this before, but just as a reminder, it's the export to a PDF file. Click on the 3 dots up in the top right-hand corner of your app menu, the 3 dots. When you do that, that gives you the export to a PDF option. I have to click File and Export to a PDF from my menu. But here's one thing that you have to do so that it prints it up really nice. You have to turn on Fit Each Sheet to a single page. So now each of these 5 tabs will become their own standalone page in the PDF file. So that is important for you to do. Then you click Next, and where do you want to send it? You could save it like it's kind of showing me to any location on my device, in the cloud, maybe iCloud, maybe Dropbox, Google Drive. So that's typically what you do. But you're in the habit of sending your PDF files to Notability. And then from there, you can use the Canvas app to turn it in kind of easily. But be aware that the Canvas app will often fail when you try to turn in things from Notability to Canvas. Double check that it worked properly if you're going to do it that way. Typically, you save it to cloud storage, and then at the Canvas website, you attach the file and you just drill down into your file storage and attach it. So the 2 ways of turning in work with Canvas. Okay, so now you know how to do that, and you could check it out to make sure that it looks right. But let's click over here to the textbook 2.4. And we're going to talk a little bit here about what is an acid and a base. Can I get a volunteer to read these 2 short paragraphs? Volunteer, or I'll call on somebody? What's that?
I can't read half those words or else.
All right, well, That's how you learn, by getting in the game. You want to read it? Yeah, thanks.
In liquid, a very small percentage of water molecules, small percentage of water molecules dissociate or break apart into hydrogen ions and hydroxide ions.
Good.
These ions are very reactive and changes in their concentrations can drastically affect the cell's proteins and other complex molecules. Some chemical compounds contribute additional H plus to an additional to an aqueous solution, whereas others remove H plus from a substance that donates hydrogen ions to solutions is called an acid. An example of a strong acid is hydrochloric acid, HCl. The acid in a gastric juice in your stomach. An acid solution has a higher concentration of H plus than HO negative.
All right, we almost have the whole story. Let's just go over a couple more. How about another volunteer? Thanks.
A base is a substance that
Thanks, Anthony.
increases the hydrogen ion concentration of a solution. Some bases, such as sodium hydroxide, do this by donating OH negative. The OH negative combines with the H positive to form H2O, thus reducing the H positive concentration. Sodium hydroxide is a common ingredient in oven cleaners. Other bases accept H positive ions from solutions, resulting in a higher OH negative concentration.
So we use the pH scale to describe how much of an acid there is or a base. The scale goes from 0 to 14 with 7 smack in the middle. And it also tells you that each number is a 10-fold increase. So going from a 5 to a 6 is 10 times stronger. 7 is the middle. 7 is neutral, it's pure water. Going lower than 7, it becomes an acid. Going higher than that, and it becomes a base. So take a look at the chart on the right-hand side. So pure water is 7. Going from a 7 to an 8, how much of an increase is that? 10 times stronger. How much stronger is it to go from 7 to 9? 100. Going from 7 to 8 is 10. Going from 8 to 9 is 10. That's 10 times 10. Going from 7 to 10 is how many? It's moving 3 numbers. It's 1,000 times stronger. So when you get down here to 14, 13, and 14, they're really strong bases. If you've ever dealt with household bleach cleaning or oven cleaner, you're going to wear gloves because within a matter of seconds, you'll feel it burning your skin. So a strong base is deadly as a strong acid. If you look at the other side, this is what battery acid is. You have to be careful dealing with big batteries like a car battery. Usually old batteries are leaky and they've got like acid that has leaked out of some of the seals and stuff. So you have to be careful of grabbing that big car battery when you're lifting it out. Not only is it about 30 pounds, but it's really got a lot of acid around it. So you got to be careful around strong acids and bases. But then some of these look like household things you'd find in your refrigerator. Lemon juice and Coca-Cola. Did you know that some people have demonstrated that you can pour a cola like in a really nasty stained thing like toilet, and it will clean it. So you got to imagine what drinking a lot of soda can do to you. Yeah, cleans out your pipes, right? No. So look at rainwater. You've heard of acid rain? Well, rain up in the clouds, that's pure. It's 7. But as it comes through the atmosphere and it's hitting pollution, it's picking up chemicals that turn it into a slight acid. So you've seen some people's older cars, they don't keep wax on it and keep it clean. All that accumulation starts to eat away the paint, and now it goes down to like the metal. So acid rain, can do some damage to stuff. Where is human blood pH? Is it at 7?
8.
It's at
It's in between 8 and 7.
7 and 8. 7.5. I think they say it's 7.4. So our tears are slightly basic. Our blood is slightly basic. Our biochemistry needs to be at a pH of slightly basic, just off of neutral. So not only do we have to have the right amount of water and nutrients in our body, but we also have to be at the right temperature and pH. Otherwise, your chemistry doesn't quite work. And that can lead to a lot of problems. They have shown that people that are battling cancer, their pH balance is off. They're usually acidic. They're usually at like 6 or 6.5. So you've heard of things like smart water, alkaline water. Alkaline means basic. Alkaline and base mean the same thing. So it's kind of a big marketing thing. And some people really try to watch the amount of acidic juices that we take. But there isn't an issue where you're going to swing your pH balance in your body. If like you just had lemon juice, it's not going to make your blood pH swing down to like 4 or something like that. So take a look at this paragraph. There's something called a buffer. That is a chemical that our body will put into the bloodstream. If our pH balance is getting off kilter, it will put a buffer in there, which is going to keep it really close to that 7.4 range so that it doesn't swing much. If you guys have seen those beautiful saltwater aquariums, and maybe you have it at home or seen one, open up the drawer underneath and they've got all these chemicals that they have to keep adding to the fish tank. And one of them is a buffer so that the pH balance stays at about 7.6 or something. So you can see seawater is pretty close to human blood. So the fish also need that. But if you're not careful in keeping the water balanced, now all of the uneaten fish food and the excrement that the fish are giving off, it starts to add all these nitrates to the water. It's going to make algae bloom, which creates the pH balance to swing. So you add buffers to it, and it tries to keep it more in balance. So our brain is like a computer that constantly monitors our blood. And if pH is getting off, it will put some buffers in there from some of our glands. So that's kind of cool, huh? So that's some of the background from our textbook. And now if you click on the background up here at the top, the background from the lab, talks about some of the same thing, but you need to come back here and read about pH in your skin and pH in shampoos like baby shampoo and tearless shampoo. When it stings your eyes, it's not the right pH balance of like 7.4. So baby shampoos will try to be close, be right on the 7.4 so it doesn't hurt young kids. And then I mentioned about acid rain too. So now if you look at the conclusion questions, it's got some down here about how does non-tearing shampoo work? What's the pH of rainwater? So it's in the background information that we are just pointing out. Take a look at the procedure now, and this tab here has an experimental question for this lab on, I have 9 different household common liquids. Some are detergents, some are food. And you're going to determine the pH value using what you see at your table, these little strips of paper. They're called pH test paper. You can buy something like this over at the drugstore. Some people have to monitor their pH balance and their glucose balance and other things. So it's just another method of testing your biochemistry. And so with every experiment, you've got a hypothesis. Well, using the pH test paper, we're going to be able to determine what the pH value of this. But let's take our best guess at what they are before you even start. So if you click over here to the data table itself, Why don't you give it a try? So you see this first column? Without looking at the data table that it just showed you, you're not going to be graded on what your guess is. But why don't you guess right now what the pH value is going to be before we test it? So take a moment. Take a moment to do that, okay? Fill in the hypothesis column in the data tab. If you think it's an acid, you're going to write down a number less than 7. Just your guess. These are my guesses. They're not necessarily right. And I'm not going to finish that. Zero to 6.9 for acids, with 1 and 0 being a strong acid. 7.1 to 14 as a base, with 14 being the strongest base. So the lowest number and the highest number are strong. In the middle is 7.
What's antacid?
Then I want to show you one more thing over here at the textbook tab. The first paragraph that Anthony read said, water can disassociate. Right here. That means break apart. Well, I think you all know what the formula for water is. How many hydrogens?
2.
1, 2. I could have written it H2O, but I said HOH. Same thing. So water breaks apart. This guy becomes H plus. This one becomes OH minus. Remember in the last class, I was having you understand what makes a positive ion? So this right here, what happened in order for it to become positive? It now is H plus, but it started off as H neutral. So how does something become positive? Did it gain a proton? Or did it lose an electron? It's all about the electrons. Remember, it's a shell game with electrons. Chemistry is about electrons. The protons don't change. So it used to have 1 negative electron. Guess where it went? How does something become negative? It has 1 extra electron. So the electron that was right here, I'll put it in red. There used to be an electron right there, but then it goes over here and becomes that extra electron right there. That's dissociation. Water splits apart. It says that it happens kind of naturally, a very small amount. Now, if you look over in the chart, they did a really nice job of what it looks like in solution. So here's a strong acid. So what does it have more of? H plus. If you look at neutral water, it has the same number of hydrogen ions as hydroxide ions. They're balanced. How do you neutralize an acid of 6? An acid of 6 strength? That's 1 away from the middle, right? Guess what you have to add to it? Would it be the same amount of 8 or the same amount of 9? If you're going 1 notch above to make an acid, how would you neutralize that acid? You would go 1 notch below 7. So to neutralize a 6, you add an 8. To neutralize a 5, you add 9 in the same equal concentrations. So take a look at a base. A base is OH minus. Aren't acids and bases just water broken up into their ions? If you have more H plus, hydrogen ions, that's an acid. OH minus, why are they both deadly? Why is this really going to hurt you? What's the problem with H plus? It's missing an electron. So you pour this acid on you, not necessarily lemon, but like battery acid, and it will rip the electrons out of your other molecules. It'll steal it and break apart your cells and tissues. Similarly, over here with the base, all these extra electrons are going to go somewhere. They're going to leave the hydroxide and go into your cells and disrupt your cells. Okay, good timing if you can't wait. So let's take a look at the data over here, and I want to show you what you're going to do. The directions are right here. We've already completed the hypothesis column. This is column A, this is column B, and column C. If you don't see these letters, you tap into the table, and then it makes all the rows and the columns visible. Record the pH value of the object in column D. So right here is column D. How do you do that? The directions right here, we'll go over them. You have a test plate at your table. It is labeled 1 through 4 and A, B, and C. So you're playing Battleship. A1 is in the back left, A2, so just kind of follow the coordinates. Right here is where they go in the wells. Along here I have everything has a number on it. So B2 is going to go into the B2 well right here. And notice that it's got a green marker and a green dropper. So don't mix them up. Keep the same color dropper in the same color container so it doesn't contaminate. It'll take a pretty good amount of liquid, but don't put it right to the top because you're going to fill it up here and then you're going to walk back to your table. So you don't want any liquid to splash and go into another well. So we can have people loaded up in the front here, and you can also come back here so that we could accommodate at least half the class at one time. And so you're going to fill your entire plate. You're going to have 9 liquids in there. Then you're going to pick up a piece of pH paper with the forceps, the tweezers. So at your table you have a Petri dish. Open it up, and instead of touching it with your finger, you may have some oils on there which would react with the pH paper. So you pick it up like that. Dip it into the well just for a few seconds. The water soaks up into here and changes colors. You have a color matching at your table, and so you just see what color it is, and it will tell you what number that color represents. And that is what you write down in the table. So the data table is already there. You're adding that, and you're going to make a chart. You're going to make a bar chart. And you're going to complete the conclusion questions, and then you're going to export this when it's all done and save it as a PDF file. So back over here to the data. You're going to write in some numbers. I'm just going to type in some random numbers just to show you what it looks like. So you're graded on the accuracy of your results. So now you have it in there. We just did record the pH value in this column with all the test strips. And now determine the pH type and record it as an acid, base, or neutral right here. Is this pH value of 9 an acid, base, or neutral? What is it? It's a base. It also says that you can specify that it's a weak or a strong base or a strong acid, so you could do that. Remember, they're weak if they're close to 7. So 9 would be a weak base. 3 would be a strong acid. An 8 would be a weak base, and so on, okay? So then you're ready for sorting the table. Select the C column and then the column actions to sort ascending. You can practice that right now so that you know how to do this. You tap on the letter, and then it pops up a menu, and then you hit column actions. And then you select sort ascending. So my menu is just a little different. Sort ascending, and boom, it changed everything in numeric order. And it moved the entire row with it when it sorted. So now you can see that the location of the test plate, which used to be A1, A2, A3, and so on, now is out of order because we sorted it by this column. So if you're dealing with thousands of records, you can sort everything in an instant with one command. So spreadsheets are really powerful in analyzing data. Then we're down here to number 5, and you are going to make a 3D bar chart. So you're going to select the C column, and it actually should read the D column. That didn't get fixed, did it? So I'll fix that. So we're sorting by D, not by C. You want to make a change to that? If you need to, you would click down here and you would say this is the D column that you're sorting by. So you tap into the table, you click the letter D, you do column actions, and then you select the chart. So now you created a chart. And look, it's in ascending order. It looks better than this. So you can't see all the x-axis values because it didn't have room for it all. So the directions say grab the dot and expand this chart so they all show up. There might even be a way of turning it from a horizontal label to a vertical label, which is nice and handy. I just haven't had the chance to find where that is yet. Then the directions say make sure that you create a title. We're on number 7. Create a title and an axis label for your X and Y axes. So remember, when you're selected on the table, you click on the paintbrush, which is called the format. You can turn on the title. You can also turn on value axis labels. Click on the axis. I'll turn on the axis names for the X and the Y. Don't leave it at this. Now you double tap on that and you call this objects. And over here you're going to tap and say pH value. You see how the legend and the title overlap each other? You can grab that and you can move it out of the way. The title would then be pH value of various liquids. And pH is spelled lowercase letter p and uppercase letter h. It stands for the power of hydrogen, because that's what acids are, right? It's a measure of the hydrogen ions and hydroxide ions. Then you would double click over here and say that this is objects. The directions also say, see if you can add the value axis labels. And right here, I'm going to add the number, and you can put it on the top, you can put it on the outside. Make sure it doesn't overlap stuff so you can make it so that it's good and readable, maybe at the bottom. So that's dummy data, right? I just typed it in just to kind of show you how to format your data table. But once you got the hang of it, it's easy to make data tables and charts. It's very professional. The directions right here, number 9, say, hey, you don't need these directions afterwards. So once you tap on it, delete it, and then just move things around so that it's a little cleaner. Then when you're ready to export this, you are going to hit the 3 dots and you are going to export this as a PDF file when you're all done. And you are exporting it using fit each sheet to a single page. And you can, on your iPad, send it right over to Notability if you'd like to take a look at it. But do all your work here in Numbers app. Don't worry about doing anything over at Notability. You're just using Notability if you need to because you like to turn in work through Notability using the Canvas app. Any questions? So you can have 2 lab groups per table, but everyone is doing their own spreadsheet. So there's a setup for 4 people to work together to collect your pHs.